Okay, we're going to continue our talk here on DNA packaging, and like I said, this video is going to be on gel electrophoresis and how the actual structure and shape of the DNA can affect what the results of a gel electrophoresis test might look like. So before we start, we want to look at what exactly is gel electrophoresis. So the gel that you actually use is called agarose gel, and it's a uncharged polysaccharide that's purified from agar of red algae, and red algae is just seaweed, so you can just write gel from seaweed. And it has three different kind of states. So its first state is its kind of solution state, which is just this free kind of molecule. It's not very tightly wound or anything. It's not really in a gel form yet. Then if you heat it up to 100 degrees Celsius, it will take this kind of coiled shape with one another, mixing in, kind of coiling around and forming this type of weird shape with a pore in the middle. And then after a little bit of aging, you would reach this tightly wound gel that has pores. It might be shaped like this. And these are the actual pores that the DNA will be traveling through in the gel tray. So to prepare the agarose gel, you first of all need a gel tray like this one right here, and you would prepare barriers that would retain this gel inside of the tray that you're using. You would pour the molten gel into the tray and insert a comb right here, so there would be little holes that you could just get online. Like the tray would probably come with the comb would come with the tray that you're using, and then you insert that in and let it harden. So then when you take the comb out, there would be these little holes, and that's where you're actually going to inject your samples. So if we're looking at the actual structure of the gel electrophoresis tray, it is going to be, it's an electric field. The reason why the DNA travels down away from this side is because DNA at around a neutral pH has a negative charge. So negatives do not want to interact with negatives. So they're going to try and get as far away as possible. And if there were no gel, they would move right across. But since there's gel, they're going to have some issues moving across and trying to get through these little pores. So let's change the color here and look at, well, actually, first of all, I just want to label the different sides of the gel electrophoresis tray. So the anode here is the positive end, so anode, and this end is the cathode. So DNA obviously is going to try and get away from the negative charge. Shorter molecules tend to move farther down than longer molecules, which I'm going to draw in a second. But what also affects it is, like I described in the last video, is the actual shape and structure of the DNA. So a linear molecule, if you look at a linear molecule that's trying to get through this pore, so I'll draw this thing again right here, a little bit smaller. So you have this little pore right there in between the agarose gel, and then you have this DNA molecule that is linear. So let's just draw this, and this is your double-stranded DNA molecule. So obviously if it was trying to go this way, it wouldn't fit, it would just keep on hitting. But if it's traveling kind of like in this weird, like moving back and forth, eventually it's going to go directly through this pore and be able to move a little bit farther. And the shorter that it is, the easier it will be to eventually get that linear horizontal shape and go through the pore. The longer it is, the harder it'll be to do that, so that's why shorter molecules move further. So now we're going to move on to the second shape, which is the relaxed circle. So like this kind of shape. This one is going to move the least out of every molecule because it's going to have a tough time getting through the pore because it's just kind of a big blob that's all over the place. There's no real orientation that's going to allow it to pass through. And the last shape that I described is the relaxed, or the supercoiled, sorry. So that's this kind of shape where it's all coiled up. And once again, you have your shape here. So this one's going to move the second most out of all the different shapes. So if we draw this here where we have your number, let's do this in pink. This is your number 1, right here. Let's just draw this right here. This is 1, this is 2, and this is 3. So here is 2, and here is 3. This one is going to move the farthest, so you might have like 10 kpb, and kpb is just the effective separation range of the linear DNA. And then for this one, it's going to move the least, so you might have 2 kpb, and then the supercoiled is going to move pretty far as well. So let's say that this is 7.5 kpb. So you can see here that if you were trying to figure out, first of all, the shape of a molecule, how many base pairs it had in it, how big a molecule was, you could use the same type of experiment using proteins, so you could find out the size of the proteins. And you know, gel electrophoresis is a huge experiment that's used every day by scientists in a lot of different fields to determine the size and shape of molecular molecules. So that's it for this video. In the next video, I'm going to talk about nitrocellulose membrane and Western blotting, which is another common experiment that is used in genetics and other scientific research.